to the light-independent reactions of photosynthesis. If you recall, photosynthesis is broken down into two major reactions. Okay, one is light-dependent. Those occur in the thylakoid membranes, and they make ATP, NADPH, which is a reduced coenzyme, has energy, and of course oxygen. Okay, that occurs in the thylakoid membranes. Okay, the light-independent, sometimes called the Calvin pathway, occurs in the stroma. And the stroma is the space between the thylakoid membranes and the outer membranes of the chloroplast. Okay, chloroplasts have three membranes: outer, inner, and then the thylakoid. So the stroma is where this light-independent reactions. This is crucial because this is where carbon is fixed. And so they carbon fixation. Okay, so this is where we're going to actually make glucose, galactose, and all the sugars and organic molecules. Okay. So it's a cycle, Calvin cycle. So it starts over here. With a molecule called ribulose biphosphate. Okay, these circles represent carbon atoms. Okay, so it's simplified. Okay. So ribulose biphosphate, which means it's got phosphates on the ends here. Okay, and there's 6 of them. Start with 6. Okay, so if you want to keep track, that's 6 times 5 carbons, so there's a total of 30 carbons there. The first thing that happens is that carbon gets fixed by this enzyme called Rubisco. Okay, that's short for ribulose biphosphate carboxylase. Okay, so that's a lot, so they just shorten that to Rubisco. Okay, so you add 6 carbons, okay, that comes in through the stomata of the plant, okay, and they combine. And then that does, now you have 36 carbons to account for. There's a quick breakdown in the middle, so now you actually end up with 12 3-carbon units, okay, which was, they're called phosphoglycerates. Okay. You got a 1-phosphate group attached here, so you have 12 of them. 12 times 3 is 36, so you have your 36 carbons to count for. And then here's where you start using some of that energy, okay, in these coupled reactions. Okay, so of course this is losing energy, ATP to ADP. You're losing some energy, okay, but then it's gaining it down here. So see, in this case you added a phosphate group. Okay, that's got some energy within it. Okay, so we say that is a coupled reaction. So you get down here, then you get one more coupled reaction where you break down some NADPH, so it's losing energy. So what you end up with down here is a molecule called phosphoglyceraldehyde, okay, which is, there's 12 of them, there's 3 carbons there. Okay, so we still have our 36, but now it's energized. Okay. 2 of those break off here, okay, out of the system, and they make sugars. Okay, so 2 of those can make hexose sugar, okay, 6 carbon sugars, okay, like glucose or galactose. Okay, that leaves you with 30 carbons now. Okay, so it rearranges. These are all enzymatically controlled reactions. Okay, so this is somewhat simplified, but there's a series of reactions. You end up with a 1-phosphate. Okay, so it's like ribulose phosphate. Okay, so you have these 5 carbons here. Okay, there's 10 of them. Okay, no, that's not right. 12. You end up with 6 of those. Excuse me. Okay, so you have 6 there. Okay, 6 times 5 is 30. Okay, and then you use up one more ATP. Okay, and you add a phosphate group. Okay, so now you end up with biphosphate. Now we're back to the start with ribulose biphosphate. All right. So of course, it's just like it keeps turning and turning as long as you get carbon dioxide coming in. Generally, carbon dioxide would be the limiting factor. Okay, so reaction, everything else is there. Okay, but of course CO2 is generally the limiting factor. Of course, if you didn't have ATP or NADPH, you wouldn't be able to go either. Okay, but that is the light-independent reactions. Now, you can have a bit of a problem though, okay, because sometimes in hot, dry climates, you get you have to close up that stomata, because that stomata is where the underside of these leaves. That's where these openings where the CO2 can come in. Okay, but if you leave those open, especially hot and dry, water leaves. Okay, so plants become dehydrated quite easily. So adapted by the plant is to close those up. Okay. The problem when you close them up, you have this thing called photorespiration occurs. So here's what happens during C3. You have CO2 with ribulose biphosphate, and you make glucose. Okay, here's the key enzyme is for fixation is rubisco. I mentioned that before. Okay. But the problem with rubisco is it also catalyzes another reaction. Generally, enzymes just do one reaction, but this one actually catalyzes oxygen with ribulose biphosphate. Okay, oxygen is quite reactive, as you know. Okay, and when that happens, you produce carbon dioxide and 4 carbons. Okay? So it's actually counter to photosynthesis because now you're producing more CO2 and you're competing for this reaction of photosynthesis. So it can really drop down the efficiency of photosynthesis in making of the sugars and oxygen. Okay, so hot, dry climates, when those stomata close, decreases the amount of CO2. This comes in, increases the oxygen, and that favors photorespiration. And remember, drive the reaction, you got a lot of oxygen, it's going to favor this reaction. You have less CO2, it's going to favor the photorespiration, which makes photosynthesis much less efficient. Okay, so there are adaptations that plants have made. Okay, so there's 2 types. There's C4 plants and there's CAM plants. So I'm just going to talk about C4 plants here. All right, use a different enzyme. It's called PEP carboxylase, and it's got a higher affinity for CO2, which means that it can pick up CO2 when CO2 is at lower levels, okay, and it picks it up quicker. Okay? So the problem, though, is it adds some more steps, okay, so it's not quite as efficient, okay, as C3 pathway. Actually, C3 pathway is also actually ultimately used, but it adds some steps, and so energetically it's not as efficient. Okay? But in hot, dry climates, it is favored. Okay? So CO2 it attaches into this PEP, phosphoenolpyruvate, okay, and it makes this thing called alloxoacetic acid or alloxoacetate. Okay? So 3 carbon molecule here, add CO2, now you have 4 carbons. Okay? It makes this molecule called malate. This occurs in the outer leaf cells, and this brings it in, though, transports it into the inner cells where the CO2 can be released to the Calvin cycle. So now it can go into this Calvin cycle. Okay? And then the malate is turned into pyruvate, which is 3 carbon sugar, and then that turns into phosphoenolpyruvate, and then it continue that cycle. So it adds an extra carbon, which is it costs a little bit, but in hot, dry climates it is favored. So that's the C4 pathway, and that does it for this